Thank you very much for coming. Before anything, I have to make an unfortunate announcement. The talk following the one we are in right now won't happen for some reasons, for life reasons, which means that you don't have to jump from the talk we are in right now to the next one. But now, let's continue. Our next speaker is Dr. Tiago Simões. Dr. Simões' research interests include combining data from living and extinct species, as well as morphological and molecular data to investigate deep time problems in reptile evolution. His talk is titled Modeling Morphological Data and the Ongoing Evolution in Deep Time Phenotypic Macroevolution. So, Dr. Simões, thank you very much for being here. You may start whenever you are ready.
And hopefully everyone can see my screen being shared now.
Yes.
Excellent. So, let's start. So, thank you again for the invitation. It's like a pleasure to be in the symposium with so many cool participants and so many interesting and diverse topics in phenotypic evolution and also the other topics for the conference. Um, so, today I'm going to be talking a little bit of the research I've been doing over the recent years and the methodological approaches I've been taking during those years as well. And for a brief start, I would like to show, briefly compare what we usually use for macroevolutionary inferences of phenotypic data. So quite commonly, we are used to the traditional approach of using phylogenetic comparative methods, where we start by getting our phylogenetic data matrix, and from that getting a time-calibrated phylogeny, hopefully with fossil data to help us assess evolution through deep time. And separately, we have trait data, such as body mass data or any other kind of variable, usually a continuous variable. And we combine the two of them in our favorite R package to assess macroevolution across different timescales, from which we can eventually obtain different values for character evolutionary rates or diversification rates using whatever is the best fit modeling model for each particular trait that we're interested on. So this is a kind of what we call a posteriori approach of assessing such patterns of evolution subsequent to phylogenetic estimates. In more recent years, there's been considerable expansion on a different approach, which is essentially combining and doing everything into a single analysis by integrating in which we can integrate the tree topology, so the structure of the tree itself, the divergence times, branch lengths, and other parameters of interest, such as the character rate of evolution, how that rate of evolution changes across different branches in the phylogeny, and other aspects of the diversification process, such as the speciation rate, the extinction rate, and the fossil recovery rate. And especially in the last 5 years, we've been seeing lots of advances in models for such approaches. And to give a brief comparison with the traditional PCM approach, we essentially have our data set, so either a molecular or morphological data set or both. We can also have continuous data included there. We eventually implement those into evolutionary tree packages using Bayesian inference, such as Beast, RevBayes, or Mr. Bayes. And in those packages, essentially, we are trying to estimate the joint probability of all of those parameters combined, the tree topologies, the branch lengths, evolution rates, and etc. So many that it becomes multidimensional, and we can't represent in just 3 dimensions, but essentially in a very simplistic representation would be something like this, from which we can extract our posterior estimates of not only the tree topologies, but also all of those different parameters in their optimal configuration. And eventually we can essentially get our summary trees and map how those character parameters or diversification parameters have changed across time. Most commonly, we see that for divergence time estimates and the errors, the potential error bars around those estimates, but you can also do that for rates of evolution, for instance. And some of the main developments over the past decade that we've had is on the development of different kinds of clock models that we use in such analysis, and which essentially govern the mode of evolution associated with trait evolution here. So for instance, if we have clock models such as autocorrelated clock models, we allow essentially parameters to change gradually throughout different lineages, whereas a correlated clock models, we allow those rate values to change more drastically along neighboring branches, or we can have other parameters such as local clocks that essentially allow values to transition among different clades. So we can also assess how which one has the best fit to the data using Bayes factors and et cetera and et cetera. So basically we have many different models available now to explore how not just molecular, but also phenotypic characters change across the phylogeny, and what's the best clock model to assess rates of character evolution and divergence times. Another major advance over the past decade, and which is still ongoing major advances, is on the family of birth-death models that essentially govern the tree diversification process itself. So that includes parameters such as the speciation rate, the extinction rate, and the fossil recovery rate. And those represent the three parameters we have in our analysis, besides a bunch of additional ones that essentially are fixed, that we are not focusing so much here, but those include the fraction of extant taxa being sampled, if you have any, the probability of being removed from the process, which is essentially an epidemiological parameter, but that is essentially translating to phylogenies as the probability of sampling ancestors or not, and the starting time of the process. And classically, this FDB parameter assumes those parameters to be all constant throughout the tree in its initial implementation. But in 2014 and 16, we had the development of new approaches in which we can actually allow those parameters to change across different time beings within or across our trees. So we will have with that different parameters on speciation, extinction, and fossilization for the early part of the tree, and then those can change for the second part of the tree, a third part of the tree, and so on and so forth, depending on how many subdivisions of the tree we are allowed to make and that we input into our analysis. Of course, that increases the number of free parameters. So the more parameters we have, the higher the risk of over-parameterization, which is especially a higher risk when you have morphological data, because we have low amounts of data typically for morphological characters. So it's always important to balance these amount of parameters with the amount of data you have available. So definitely when setting up such analysis, it's quite important to keep this particular aspect in mind and avoid using too many parameters. One of the other things that has been quite important is the understanding that we may have very different behaviors of such analysis depending on the strategy that we use to sample modern taxa. So for instance, we can assume a random sampling of both fossil and extant taxa, or a diversity sampling strategy, which is in fact how most phylogenies using extant taxa are sampled. So most such phylogenetic data sets, they have a taxonomical representation that is proportional with the diversity of each particular clade. So, and quite importantly, we know that such different assumptions have a huge impact on divergence time estimates and other estimates of macroevolution parameters. So it's quite important to take those into account, besides the sampling or not of fossils as potential ancestors during the process. And regarding this particular later aspect, I recently assessed the impact of that using morphological data sets, using this particular phylogeny, this new data set that I built for Sphenodontian reptiles, so essentially the modern tuatara of New Zealand and all of its extinct lineages, extinct relatives. And you don't need to know anything about Sphenodontian phylogeny. The really important thing to know here is that we have here multiple trees represented on top of each other from different analyses and using different models. And we have here divergence times being depicted, and the chronological time on the bottom. So as you can see, the nodes closer to the tips, they have very low difference, very little variance on divergence time estimate for those groups, usually based depending on model implementation. But as we get closer to the root, we see huge variations on that, which is already known for molecular data, but it seems to definitely impact morphological data as well. But interestingly, when we apply the most commonly implemented models, which assume fossils to be ancestors in the tree when using the FBD model or and implementing the best fit clock model to this particular analysis, and the classical assumption of the FBD process, where all of those diversification aspects are constant throughout the tree, that's when we get the oldest possible divergence time estimates closer to the root of the tree, and which are in strong disagreement with the data from the fossil record that we have available. However, if we relax the assumption of fossils being actually, if we constrain the assumption, so if we remove fossils from being ancestors and we make them to be exclusively tips, and there are many reasons why we would assume so for this particular data set, it's a very small sampling of taxa over 200 million years. So the probability of sampling direct samples, ancestors and descendants, it's quite minimal here. So if we don't assume that, we actually get much younger divergence times and in closer agreement with the fossil record. And if we, regardless of the clock model we choose, both the best fit and the not best fit clock, in the least fit clock model. And if we, along with that, we also implement the FBD parameters being variable across time. So the Skyline FBD model, we get even younger and more conservative divergence times, which are in stronger agreement with the fossil record. And so it seems that incorrect modeling can definitely overestimate divergence times using morphological data just as much as with molecular data, but we can easily correct for that by knowing exactly what the models do to each kind of to our data set. And so it's definitely important to test different models for the data set we have at hand. And I'd like to show some of the immediate applications and results that we obtained by using such models for macroevolution analysis over the recent years. One of the most common approaches is to estimate divergence times, which people are more familiar with from molecular data and also morphological data more recently using morphological clocks. And for instance, in an example of this, in a recent paper in 2018, I estimated that the origin of the major groups of reptiles, so the deepest branches in the reptile tree of life, some of them actually originate in the Permian period, so beyond 252 million years ago, instead of actually originating in the Triassic between 252 and 200 million years ago, when historically paleontologists believed that those groups had originated. And instead, we show that despite the oldest known fossils being from the Triassic, in fact, that entails by those models, by applying relaxed clocks, that the origination times of those groups would be much earlier before the Permian Triassic extinction. And it's only during the Triassic that they expand in diversity and abundance so much that we actually can sample them in the fossil record and find them in the Triassic to begin with. Besides that, we also have applications to understand the rate of character evolution. And one of the first approaches to that was done by Mike Lee and colleagues in 2014 to understand the rate of evolution during the dinosaur bird transition during the Jurassic and the Cretaceous. And what they found is that there is accelerated rates of evolution in the lineage leading to birds, and exceptionally high rates of evolution leading to that, which actually conforms to the classical theory of new body plans emerging at high rates of evolution, first developed by George Gaylord Simpson in the 40s and early 50s. Beyond that, more recently, I also estimated such rates of character evolution using the data set of that phylogeny I showed from 2018, but expanding the sampling of both recent and fossil species with special interest on squamate reptiles, my main group of interest. So all groups of lizards and snakes, and and other groups of the diapsy reptiles represented here, mostly extinct or the early members of groups that live to the present time. And by doing that and integrating that with molecular data, could estimate the rates of phenotypic and molecular rates of evolution throughout the squamate tree of life. And essentially what we obtain here is values of and distribution of rates of both morphological and molecular evolution, which are quite similar to each other in terms of their shape, but not exactly, but they definitely do not occur on similar points on on the phylogenetic tree. So the points on the tree where we have high rates of phenotypic evolution, we tend to have low rates of molecular evolution and vice versa, displaying therefore a huge, strongly disjoint distribution of those rates across evolutionary time in squamates, at least concerning molecular protein coding loci, which I have sampled for in this molecular data set. We had subsequently implementation of estimating rates of morphological evolution based on different kinds of morphological traits. So in this paper by Patterson et al. In PNAS, they estimated differently the rates of evolution for discrete characters, meristic characters, and continuous characters. In this case, finding very similar patterns of evolution for the early evolution of trilobites during the Cambrian. And we can also estimate rates of evolution separately across an anatomical region, as also performed for this Twin data set I mentioned previously, in which we estimate rates of evolution for the skull against the mandibles and against the postcranium. And we can compare how those rates change across time. Usually they actually change similarly, but with, of course, some particular differences, such as, for instance, higher rates of evolution on the skull relative to the mandibles, inclivosaurs, and some other groups. And we can compare those rates to each other as well and see which ones have kind of stronger correlation, or at least they provide better fitting linear models. And to that extent, we can see, even though we're separating those rates apart, like estimating them independently, how much they are actually similar to each other. More recently, I've been implementing those approaches to a different problem, really outside my usual scope of research, usually focused on reptiles, now focusing on the early tetrapod evolution. So the transition from fish to early tetrapods, which has been the subject of various research papers over the past decades, and which has been estimated to have taken place during at some point between the Middle and the Late Devonian. And differently from the previous approaches in which partitioning morphological data by anatomical region is quite straightforward, in some instances like this one, it can be quite challenging by what I call the pectoral girdle problem. And that's because the elements of the pectoral girdle of most tetrapods, so the shoulder bones and the bones in the limbs, they essentially become, they're part of the pectoral girdle in tetrapods, but they're still associated with the skull bones in fish. So when we have a data set including both fish and tetrapods, to each particular partition should we assign those bones to? Well, that's a big open question. So it's interesting in this particular case and some other cases to perform a more objective assessment of morphological partitions for evolutionary rate inferences. So towards that goal, there is one available implementation in Partition Finder, a program to assess the partitions and models of molecular evolution in molecular data. But it also allows you to assess partitions for morphological data using distance matrices. So it creates an inter-character distance matrix. And from that, it estimates Euclidean distances between those characters. And we can, by using such distances, we can use a clustering algorithm such as k-means to assess which particular regions are clustering together. However, it suffers from some limitations, such as the fact that Euclidean distances are quite well known now to be severely impacted by missing data. And whenever you have a data set full of fossils like this one, that's a huge problem. And k-means are particularly susceptible to outlier biases. So I'm creating a new protocol in which we are using Gauer distances to estimate this distance matrix, which is considerably more robust to missing data than Euclidean distances, and partitioning around medoids as our clustering algorithm, which enables not only Euclidean distances, but also the Gauer distances that we're measuring here, and it's less prone to outlier biases. I also used a second test to kind of verify the first one in terms of the configuration of those partitions. And it's, in this case, a method called t-SNEEZ, which is similar to multidimensional scaling, but it's a more recent development and tested and assessed to be actually superior to traditional multidimensional scaling. And the patterns that we see with this approach here, we essentially see three major clusters. And when we color the data points here, according to the partitioning scheme we got from the previous approach using PAM, we essentially get a very similar configuration. So the colors match the clusters we see from the t-SNEs quite well. And essentially we have three major partitions here. And those partitions actually have a very close relationship with anatomical divisions, which is quite good and intuitive. Essentially, we have two cranial partitions and one postcranial partition, the first one being containing most of the cranial characters. And those spectral girdle elements, they in this case here, they clustered with the cranial partitions, with the cranial elements of the first cranial partition. So essentially, the data distribution seems to agree that they should be clustering with the cranial elements instead of the postcranial in this case. Using this approach, actually expanding the approach of estimating rates of evolution, we can also utilize those rates of evolution to measure strength of natural selection. This is something that is done for molecular data sets. And recently in 2016, Baker and colleagues actually implemented that to morphological data sets. And what they consider is that by measuring significantly accelerated regions of the tree to be undergoing positive selection and regions undergoing significantly or considerable decelerating rates of evolution to mean that they're undergoing stabilizing selection. So essentially constraining that particular phenotype. And this has been implemented in the program base traits, which assesses continuous trait evolution. But I wanted to implement that to morphological clocks, so we could also measure that for discrete characters. And the approach is essentially, it's quite similar. So not really going into all the details of that, but I can go over during the questions time. But essentially what we have here is the rate of evolution. If we find rates at branches which are equal to, which are higher than the background rates of evolution plus one standard deviation, we find those to be significantly accelerating, therefore undergoing positive selection. And if we find those rates to be less than the background rates minus one standard deviation to be significantly decelerating. And by taking that and plotting that over the data set, we can actually see which branches of the tree are actually going different selection strengths. So the result for this particular, by using the only available data set for this particular transition, we found that the group called Episthosigalians, which include transitional forms like Tiktaalik and the first known tetrapods, they are undergoing strong rates of evolution, accelerating rates of evolution relative to all other clades in this particular phylogeny, including Tristicopterids, Rhizodontids, Osteoleptids, and the earliest known Dipnomorphans. So those are all sarcopterygians, and we have tetrapods, so limbed vertebrates over here. And this is true whether you assess that over all morphological partitions as a single unit, or by actually estimating rates independently across partitions. So we always have rates higher for Episthosigalians relative to the other groups for all morphological partitions that we can assess here. When we try to see how rates are significantly faster or slower, indicative of positive selection or stabilizing selection, we find that we have rates close to background among some of the ancestral nodes, but we have really low rates indicating stabilizing selection for most of those clades, especially for the this one here, which is the cranial partition number 2 and the postcranial partition, and for two clades with the cranial partition 1. And only Episthosigalians are able to break away from those stabilizing selection and actually undergo fast rates of phenotypic evolution indicative of accelerating selection, therefore indicating that they were able to break away from those patterns only during the deployment of the tetrapod body plan during the middle to late Devonian. So in summary, we have had over the past decade, and especially over the past 5 years, fast development of new models for Bayesian evolutionary inferences using, including morphological data and the inclusion of fossil data. And this is especially important because it promotes the integration of fossil data, not only to existing morphological data sets, but also with ongoing developments with and expansion of molecular data sets, including genomic data sets. And beyond divergence time estimates, we are able to use those Bayesian approaches to actually measure the rates of morphological evolution and compare that to molecular evolution, and even calculate the strength of natural selection in completely extinct lineages, which is something that we definitely didn't think was possible until quite recently. So with that, I would like to thank all the organizers of the SBE meeting for inviting me for this symposium. If you need or would like to contact me, please, you can just shoot me an email, or even on Twitter as well, you can contact me. And to thank all the support that I had for my postdoctoral research from NSERC in Canada and the Museum of Comparative Zoology.
Before we go to the Q&A, I have some announcements. First, every guest that may have a question for Tiago, you can write the question in chat, and I will write it and I will read it shortly. The second announcement is that we will not be having the Professor Mabi talk that would be following this one, so you don't have to abandon to migrate from this link. You can stay on this Zoom room. And the third thing is that the the SBE meeting will be having a workshop on QGIS, which I recently learned is a software for geographic mapping that people use for publication and in biogeography and this sort of line of work. It's free, it is in Spanish, and it's a very nice opportunity actually. So if you're interested, go check the links that I posted on the chat, and I will repost it now. Okay. It seems like we don't have any question.
Great. So I guess it was very clear. I hope so.
It was. Okay, so if we have no questions left, I'd like to thank again Tiago Simões. Thank you very much for being here with us today, for giving this outstanding talk.
Awesome. Thank you so much for the invitation, guys. Best of luck there.
Thank you. And I think this session is ending. Thank you very much all for coming.